Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. I'm glad you're here in today's video. We have got a rather challenging subject we're going to cover, but don't worry, we're going to make it as simple as we possibly can. And the nice thing is you can rewatch this video multiple times. I want to say this from the start. Nobody expects a student, I do not expect my students to know this like an expert after watching this one time. It just doesn't work that way. When we're dealing with things that are a little bit more challenging, it's all about time, and it's all about repetition. And so don't feel like you have to know everything and know this all after watching this video the first time. Go through, watch it a few times, just take it one little concept at a time. And so in today's video, we're going to be talking about basically ATP and how we utilize ATP and how we restore it, because ATP is the energy currency inside the cell. And we're going to be talking about three major processes today: glycolysis, the transition step, and the Krebs cycle, otherwise known as a citric acid cycle, otherwise known as a tricarboxylic acid cycle, TCA. It all means the same thing. And so we're going to talk about those things. Hope you enjoy this video. All right, so the first thing, before we get into all the nitty gritty and all the details, we need to talk about ATP. ATP is adenosine triphosphate. It is the energy currency inside the cell. It's got those phosphate groups, and particularly that third phosphate is a high energy bond. And that phosphate is transferred between ATP and other compounds, and that's what actually does work inside the cell. And so you can see here that ATP is the usable stuff, but once we've used it, once we've spent that phosphate and released that energy, we're left with something far less useful, and that's ADP, adenosine diphosphate. This entire video and the next one is really centered around, once we have ADP at the bottom, how do we restore it? How do we get the phosphate group reattached so that we can use it to do more reactions? And that process is called phosphorylation. And so really that's what we're talking about today. We're talking about how to get phosphates reattached to ADP. So there are three main ways in which we can reattach this phosphate group, two of which we do, and the third one, that's going to be for a later video in plants. But the first one is called substrate level phosphorylation. In other words, what we're doing here, what you need to be thinking about when it comes to substrate level phosphorylation is we're reattaching the phosphate group, taking ADP, attaching the phosphate, restoring it to ATP, utilizing a substrate, glucose, one of its derivatives, and some enzymes. And I'm going to show you exactly what that looks like in a few minutes here. So substrate level phosphorylation, we're going to need a substrate, glucose and its derivatives, and an enzyme to reattach a phosphate group. The next one, and by far the most important one for us human beings, is called oxidative phosphorylation. And here what we're doing is something called a redox reaction. We're going to explain that more in a little bit, but we're going to be transferring electrons going through oxidation-reduction reactions to reattach phosphate groups. What you need to be thinking about right here is oxidative phosphorylation implies that we are using something called the electron transport chain to reattach the phosphate group. And I'll explain in the next video how that happens. But for right now, oxidative phosphorylation, you're thinking electron transport chain. The third type of phosphorylation, this is not something we do unless you've got some type of superhero power that I am not aware of. This is called photophosphorylation, and this involves photosynthesis, the utilization of visible light and an electron source, usually water in our plants and in algae and in cyanobacteria. And what we're doing there, what the photosynthetic organism is doing there is using visible light to reattach phosphate groups. That is something we will cover in a future video. So I mentioned that when it comes to oxidative phosphorylation, that one that's super important, that it consists of a series of redox reactions, otherwise known as oxidation-reduction reactions. And I need to take a couple seconds and explain what that means. And so this picture here is showing us a redox reaction. We have two compounds. We have compound A and we have compound B. And compound A is going to transfer electrons to compound B. That is an oxidation-reduction reaction or a redox reaction. And so what we have to understand is the terminology that we use. When compound A loses electrons, so it had electrons in the upper left-hand corner, upper right-hand corner, they're gone. That process is called oxidation. We can say that compound A has been oxidized. So anytime a compound loses electrons, it has been oxidized. Compound B, in this interaction between the two, it gained the electrons. So from right to left, you can see on the right-hand side, it didn't have the electrons. On the left-hand side, it didn't have the electrons. On the right-hand side, it does. In that process, when compound B gained the electrons, it has been reduced. Now, I know what you're thinking. These terms are stupid. They are exactly the opposite of what I see going on. How can you say that something has been reduced when it actually acquired something? I understand that. I understand the terms seem silly and backwards. In biology, our primary focus is what is happening to electrons between the compounds. However, these terms make a lot more sense when you study chemistry. And so if you really want to dig deep, go find your chemistry textbook or go start studying chemistry, and they'll tell you more about why this reaction is labeled the way it is. But for bio, we just need to understand compound A lost electrons, it has been oxidized. When a compound gains electrons, like compound B, it has been reduced. Importantly, once a compound has been reduced, we now say it has reducing power, the ability to reduce something else. And so you're going to hear that term come up here in a few minutes. All right, so before we get into the real meat of this video, I want to talk a little bit about carbohydrates, because a lot of people don't necessarily understand when we consume carbohydrates, it's really for one of two reasons. Reason number one, and the primary focus in this video today, is we need the carbohydrates to obtain energy. Now, what a lot of people don't know is that really what we're doing when we crave the energy, when we crave the carbohydrates, what we really want are the electrons and the protons that are contained within the carbs. It sounds crazy to think that way, but that's really what we want. We want the electrons and the protons. The other reason we consume carbohydrates is for the second concept here called precursor metabolites. This is a fancy term that's used to describe when we consume carbohydrates and we use the derivatives of the breakdown of glucose to make some other organic compound. Examples, amino acids. You may have heard that there are 20 amino acids. There are some essential amino acids, and there are some non-essential amino acids. Well, what does that mean? Does it mean the non-essential amino acids, we just don't need them? No, we need all 20. But what it means is for the non-essential amino acids, as long as we're consuming carbohydrates, our bodies can convert the carbs to those amino acids. For the essential amino acids, we can't do that. We have to actually consume those from a protein source. So we can use the carbs to make things like amino acids and then later proteins. We can use the carbs to make lipids. Bacteria that have cell walls can also use the carbs to make cell wall components, things like peptidoglycan and LPS, otherwise known as lipopolysaccharide, which is important for our microbiology students. So the bottom line is, for carbohydrates, you're either using them for energy or you're using them to make something else. Again, our focus from this point forward is going to be, well, if we're using them for energy, how do we do that? So you may be wondering why I have a taxi cab here. I use this taxi cab to illustrate an important point. If we go back to the previous section, we said, hey, we need the carbs to produce energy, and we really want the electrons and the protons. So in order to use the electrons and protons, we have to have a mechanism to get them from the sugars that we consume to an area in the cell, usually the electron transport chain, which we'll talk about in the next video. We have to get them from the sugars to the electron transport chain. And we're going to need electron carriers to do that. And the electron carriers, to me, are kind of like taxi cabs or Uber or Lyft, whatever. But we have two states of electron carriers. What we're looking at now, and you should make a note of this, there are three types of electron carriers that we're going to focus on. These forms are in their oxidized state. In other words, they're like an empty taxi cab. And their names are NAD+, otherwise known as nicotinamide adenine dinucleotide. I don't require my students to know that. NAD+ is perfectly fine. FAD+, and NADP+. The first two, the NAD+, the FAD+, really important for us human beings. The third one is a lot more predominant in photosynthetic organisms. That's the NAD+, which we'll talk about a little bit later. So right now, these electron carriers are in their oxidized forms. It's kind of like an empty taxi cab. It's got no passengers. It's got no luggage. In order for them to be useful, they're going to have to go pick up their passengers and luggage and become the reduced forms. And so this taxi cab has got a happy passenger in there, and that happy passenger is kind of like the electrons and protons that we need to pick up from our glucose and deliver them to the electron transport chain. And so here we have what are known as the reduced forms of electron carriers: NADH, FADH2, and NADPH. Again, NADH and FADH2 we're going to be talking about a lot in this video and in the next one. NADPH we'll talk about more when we get to photosynthesis. Now, you're also going to hear me use another term to describe electron carriers, and that is called reducing power. Remember when I said once something has gained electrons and protons, especially electrons, it now has the power to reduce something else? Well, again, here we're looking at the reduced forms of the electron carriers, and now that they're reduced, they have reducing power. They have the power to reduce something else. And so I'm going to use these two terms interchangeably. Electron carriers and reducing power for the rest of this video and the next one means the exact same thing. All right, so now that we have the players involved, we need to get into the meat of this video, and that is how are we going to extract energy from glucose. Now, I know this picture looks a little bit intimidating, and like I said at the beginning of the video, please watch this video more than once. Take it in small steps. Nobody expects you to learn this. Your professor, for my students, I don't expect them to know this the first time they are exposed to it, the first time they're trying to deal with it. So what we're looking at here is the process of glycolysis. The word literally means to split glucose. And so what we're going to do is I am not going to explain every single step of this process and all the enzymes involved. That is beyond the scope of what I want to do here. What I do, however, want to do is make sure we extract the important aspects of this process. I'm going to make it really easy for you. We'll summarize it in a little bit. So, first thing, we're starting with glucose. In the very upper part of this picture, we see glucose. Glucose is a 6-carbon sugar. We're going to be breaking this glucose down. If you look all the way at the bottom of the picture, we're going to have two. It says PYR. It's called pyruvate. So we're going to go from glucose to pyruvate, two of them. Okay, so in this process of glycolysis, well, why do we do it? What is the useful things that we are obtaining? All right, so let's take a closer look at the picture. We have glucose at the top. Now you're going to see at the very beginning, we have ATP being converted to ADP. You're like, well, wait a minute. I thought you said that this whole process was supposed to create ATP, and right here you're telling me right from the start that we're using it, we're consuming it, and that is absolutely true. And so at the very beginning of this process, it's called the energy investment phase. Sometimes you have to invest a little bit to get a return. That's what's happening here. So at the very beginning, you're going to see not only do we use one ATP, but 2 steps down, we use a second one. And so right at the beginning, before glucose splits into 2, we actually spend 2 ATP. So we're -2 right now. That's not a great deal, but it will be when we look at what happens after glucose splits in 2. And so once it splits, you'll notice that we obtain some useful stuff. First thing is the NADH, that reducing power. Now that's not immediately useful. We can't necessarily do a lot of reactions with that. However, we will be able to use it later on at the electron transport chain to generate a whole bunch of oxidative phosphorylation ATP. So the NADH, you can see we have one on the left and one on the right. So we got 2 of them. So that's good. So right now we have 2 NADH. But then look what happens. After the NADH, we actually start to generate substrate level phosphorylation ATP. And so I talked about those 3 types of phosphorylation. This is an example of substrate level phosphorylation using the substrate and enzymes not shown here to reattach phosphate groups. You can see that we've got 2 ATP on the left-hand side and 2 on the right-hand side. So we got 4. If we account for the 2 that we lost at the top, we actually gained a net total of 2 ATP. Okay, I know I just said a lot of things. Let's go ahead and summarize it. Let's make it easy for us. And so here we go. This is a summary of the things that I expect my students to know, and I think it'll be really useful for you too. And so for glycolysis, we started with glucose, 1 glucose. The SLP is substrate level phosphorylation ATP. We got 2 of them. RP stands for reducing power. We gained 2 NADH. Trust me right now, if you look at that picture, you're not going to see any CO2 being generated. I'll explain more why that's important in a few minutes. We ended with the 2-pyruvate at the very bottom. And the only thing you really can't gain from this picture is where it's happening. So I'll just tell you that it's happening in the cytoplasm of a cell. Okay, that's glycolysis. That's what you need to know about, at least to me, that's the important things of glycolysis. Now, let's move on, because the next thing, there's 3 steps. The next one is called transition. I know this picture looks like there's a lot going on, but with the transition step, I'm here to tell you it's actually not, because transition step is, you see the pyruvate at the very top? It's pyruvate to the acetyl-CoA. It's just that one little line before the circle. So the transition step is pyruvate to acetyl-CoA. Now, we do have to be careful here, because that summary that I just showed you, that is all the ATP and reducing power per glucose. Now, I want to back up for a second here, if we can. So let's go back to our previous picture. You see at the bottom, we had 2-pyruvate, correct? We went glucose to 2-pyruvate. However, when we look at this next picture, we're only seeing 1-pyruvate, because what's the point of showing you the exact same thing twice? But we do have a second pyruvate going through this exact same process. And so the numbers that we obtain for ATP and reducing power from this picture, we have to double everything to account for the second pyruvate that's not being shown here. So pyruvate to acetyl-CoA, we don't see any ATP being generated here, so no substrate level phosphorylation. We do, however, have reducing power. We see NAD plus being converted to NADH. We see one of them being converted, but again, we have to double them, so we've got 2. We also see that there is some CO2 being released. We see 1 CO2 being released. We have to double that because we have a second one from the other pyruvate. So again, let's summarize everything. And so now in the transition step, we started with 2-pyruvate, which is what we ended with in glycolysis. There was no substrate level ATP being produced, so that's kind of sad. But we did get 2 NADH, and we're going to see in the next video, NADH, that reducing power is everything. That is really important stuff. We got 2 of them there. We did generate 2 carbon dioxide, or CO2. We produced 2 acetyl-CoA, because there would have been 1 happening for each pyruvate. And then where in the cell this is happening, this is where things get a little bit weird. In prokaryotic or bacterial cells, this is happening in the cytoplasm, because they don't have mitochondria, they don't have membrane-bound organelles. But in eukaryotic cells like ours, this process has now moved into the mitochondria. So at this point, we've got 2 of the 3 done. It's not that bad. Let's do the one last part, and that is back to this picture. We got to deal with the circle, otherwise known as the Krebs cycle, citric acid cycle, or TCA. Okay. So here, we're starting with acetyl-CoA. That's what we ended with in transition. We're going to see that acetyl-CoA combines with that oxaloacetate, and we get a 6-carbon citrate. Now what's going to happen is that acetyl-CoA is going to go through this process, and we're going to extract all the remaining electrons and protons that we can from those 2 carbons. And so again, what we're seeing here is, if you look at the upper right-hand corner of the actual Krebs cycle, you can see that we've produced an NADH. And then to the lower right-hand side, you can see there's another one, so that's 2. And on the upper left-hand side, there's a 3rd one, so that's 3. And again, we have to double it because there's a second acetyl-CoA, so we've got 6 of them, 6 NADH. Look at the bottom of the Krebs cycle. You will see that we have 1 substrate level ATP, so substrate level phosphorylation has happened there. Again, we have to double it, so we have a total of 2. And then there's something new. The something new is the FADH2. We've produced that on the lower left-hand side. We see 1 of them. Again, we're going to double it for the other acetyl-CoA that we are not seeing. So we got 2 FADH2. You will also notice on the right-hand side of the cycle, we have 2 CO2s that are being released. Again, you double that, you wind up with 4. Okay, so let's summarize what we've seen here. So for TCA, Krebs cycle, citric acid cycle, we started with 2 acetyl-CoA. We got 2 substrate level phosphorylation ATP. Reducing power, we got 6 NADH and 2 FADH2. We got 4 CO2. We ended with what we started with, the 2-oxaloacetate. That is always just going to be part of the cycle, picking up more and more acetyl-CoA. And then the location is the same as transition. Now, I want to kind of talk about that CO2 for a second, because it's important. The reason why we tracked it is, look how many CO2 we have. We have 6 of them. How many carbons was glucose. 6. I want to clear up a misconception. You're going to be smarter for knowing this. A lot of people think that we breathe in oxygen and convert that oxygen to CO2, and that is absolutely not true. What we're hopefully learning is that the CO2 that we are all exhaling right now is actually a waste product from the carbohydrates that we consume. And so we do not breathe in oxygen and convert it to CO2. We consume carbohydrates and convert that to CO2. It is a waste product. If we were to conclude with glycolysis, transition, and TCA, what we have done is we have created 4 substrate level ATP, right? So in this process, we've got the 4 substrate level ATP. We can use that right away, right, to power reactions. We also created from those 2 steps, we now have 10 NADH and 2 FADH2. Those are going to go to the electron transport chain, which we are going to discuss in the next video. Super important. We've completely obliterated our glucose. We started with a 6-carbon molecule, and that's what we ended with. And so those, to me, are the important things that I require my students to know about glycolysis, transition, and TCA. Okay, so in the next video then, what we're going to discuss is what are we doing with the reducing power? How are we going to use that at the electron transport chain to make a mess load of ATP through oxidative phosphorylation? That video will be in the upper right-hand corner. As always, thank you so much for being here. Have a great week, everybody.